Hi, we have Aly Khan from Chicago. He's going to talk to us about digital pathology.
Thanks. Thanks for the invitation, and it's actually great to be back at CMU after many, many years when I was actually a student in Russell Schwartz's lab. So it's really a pleasure to be here again. For a long time, my work has been in computational immunology, where I've been interested in extracting immunological signals from basically a common modality of genomic sequencing data. So whether it's RNA sequencing or DNA sequencing, ChIP-seq, those types of experiments. And so with our growing appreciation of the role that the immune system plays in cancer, a new type of data modality has come into view for my work, and I want to talk about that, which is kind of these histopathology images. And so what I want to do today is I want to just quickly kind of explain what that data modality is, introduce some basic but I think important problems that that field will have in terms of incorporating it with genomic data. And then I'll try to, at least within 7 minutes, persuade you that we're making some inroads on solving some of those basic problems, but it's an exciting area that I hope you guys will agree. In the interest of time, probably the easiest way to kind of explain all this is in the context of cancer. So for a typical cancer patient who comes into an academic medical center to be treated for cancer, the patient will come in, there's be a pathology review, then a biopsy of that tumor will probably get set for sequencing. A lot of our work, and especially I think for Trey and Fabio, they've been able to show that as a community, we've developed a lot of great tools and approaches for taking genomic sequencing data across a variety of cancer types, exploiting and extracting signals from that. But what's been neglected, I would say, in kind of the way we've interacted with medicine is we've neglected the types of data that come from pathology review. So in particular, the types of data that we're neglecting is these types of images. So what this is like a section of a tumor, and it's been stained with a special kind of dye to enhance the contrast. So what you're seeing is kind of like the cellular and tissue morphology. So one question is, well, what can you do with this, and why is this even important in the context of the post-genomic era? Well, the first thing you can do is you can try to say that we know that pathology imaging occurs very early on in the staging and diagnosis of a cancer patient. So can we use some of that imaging information to actually predict something about that patient's genetics that could be therapeutically relevant? The other thing is that there hasn't been a lot of attention in how do we effectively integrate genomic data with imaging data. And so both of these are, as I mentioned, very basic problems, but they're important in the way we want to effectively utilize this data type. Of course, all of this is predicated on the belief that we can actually apply a lot of this current advances in machine learning and deep learning to histopathology images in a effective manner. So on that note, and especially in kind of as a riff on Saket's talk, there's certainly pigeons that have been trained to identify tumor versus normal. So here we have a biological animal that's able to recognize it. Clearly something interesting is going on here. So I agree, can we go back and figure out what the pigeon is doing, let alone what a pathologist is doing? Also, hopefully there's no pathologists in this room. So when I'm at Chicago, this is a very controversial slide. All right, so two key problems. The first one is, in imaging data, I'm not going to talk about typical classification. I want to talk about typical classification in the context of domain adaptation. So the idea is that when you're doing genomic sequencing and you're looking at mutations and pathways, you're often looking across cancer types. With imaging, there's a very specific problem here that I'll get into. And the second one is, because this is kind of almost clinical facing, we need to be able to interpret these models in a meaningful way. So for the first example of why domain adaptation is a salient problem for us, there is a very important biological phenotype or clinical phenotype called microsatellite instability. So this is a genomic indication where in certain cancer patients, their genome becomes highly unstable. And oftentimes, these patients are excellent candidates for immunotherapy, for cancer immunotherapies. In fact, they're such good patients that for the first time, about like 2 years now, since 2017, that the FDA approved this genomic indication. So if you take a patient's DNA and you make a classification that they're MSI high, there's an FDA approval for that patient to be treated with cancer immunotherapies. This is a really important indication. And so one thought is, rather than waiting 2.5 weeks for, even in the best case when you can get sequencing data for a cancer patient, can we make a type of prediction at the earliest time point in that patient's diagnosis and staging? So can we take an H&E image and can we classify whether or not that patient has that genomic instability or not? And that could be incredibly powerful, especially in the context of patients who might not have access to sequencing. So if you think about around the world, not every place will have an Illumina sequencer at a community or rural hospital. So in a very naive setup, what we did is we took a bunch of histopathology images and we tried to predict whether we could get MSI status. And actually, you know what? It's not bad. It actually works pretty well. And there's already papers in the field that are emergent where there's discussions about how you can take those histopathology images and start to predict genomic mutations or phenotypes. What's really interesting and striking, and if you look at that work, is that these are often techniques and algorithms that are applied to a very specific cancer type. So often it's lung cancer, colorectal cancer. Even then it's often a very specific subtype of lung cancer. We see something really interesting even in our own work, and it's reproducible in other work too. When you train a model on lung cancer and you test it, let's say, on uterine cancer, the model actually does not work very well at all. What's happening is that it's learning some sort of biased representation of this phenotype from that image, and it's taking into account, for instance, colorectal specific morphological signals, and that does not generalize well or translate well to other cancer types. Now, the claim earlier on was that microcellular instability is a genomic phenotype. It actually occurs in a variety of cancer types. So there should be some sort of invariant representation across these cancer types, even at this morphological level, that can help distinguish patients who have MSI high versus low. So we've started taking some current work in computer vision for domain adaptation that's actually proven to be quite useful. So in the naive model at the top, where you're basically just doing a simple classification of the MSI status, you could modify that architecture to have this kind of inverse gradient layer. So what you're doing is you're going to adversarially train this model where when given the cancer type, you say, look, I don't want you to know what the cancer type is. That embedding, forget about being useful for predicting a biased representation of what you've seen. Instead, just focus most of your time on MSI status. And I won't go into the details of the results, but this actually works quite well. There's a manuscript forthcoming, so the details will be there. What's nice about all of this is that we can now go back and look at this work through the lens of some of the same questions people are trying to answer in computer vision, which is when we develop these large deep learning models, how do we actually interpret what's going on? And so we've adopted guided backpropagation to look at the pixels that will lead to class activation. So here's original source images, and then here's kind of what's getting activated for specific class labels of MSI high. And the arrows are actually pointing to these small spots on these tissues where you have like a dark black circle with a white halo around it that has infiltrated into these tumors. So any guess what that might be? They're immune cells. Okay, so I would have waited longer, but I feel like I'm out of time. So the idea is that we're actually now able to train a model, look at what's invariant across these cancer types, and go back and see what's biologically meaningful. There's no shortage of questions here. Questions about what the ideal architecture is. Does this mean we need to look at neural architecture search to get a better understanding of what exactly is the right way to go about with digital pathology images? Everything we're using these days is still based on work that's been done using ImageNet data. Can we look at other types of phenotypes, not just specific mutations or collections of mutations, but perhaps networks? And then model interpretation. There's still questions about what's the best way to look at interpretation, not through the lens of what has happened in ImageNet, but maybe through pathology. All right, I'll end there, and happy to talk afterwards. Thanks.
Thank you. Are there any questions? We can take maybe one or two at most.
But is there any possible bias introduced by specific tools or specific microscope or dye that can, confound with the results?
Yeah, absolutely. What we found is that image augmentation has a very important role in all of this. So to really counteract the effect, so in our work, we've been trying to do a validation of our methods on data from a different hospital. And one of the things we've noticed is that, yeah, the luminescence of the scanner, the dyes, and everything else is going to play a role. And one way to circumvent that people in vision have been doing for other tasks, is image augmentation. So perturbing the training data to kind of fit what you might see across there. But it's an important thing, and better solutions have yet to be discovered.
Any more questions? All right, with that, let's thank the speaker again.